Hey everybody, welcome back. Now we're going to talk a little bit about the gustatory system. So we've talked about taste and the difference between taste and flavor. We've talked about why the tongue map is a lie. Now let's actually talk about the gustatory system itself. So the gustatory system basically has a variety of different outputs, as you can kind of see here. So we have our tongue. You can see different nerve fibers that will be making contact with our taste receptors, and these are divided into a few different components. That includes areas like the vagus nerve, cranial nerve X, our glossopharyngeal nerve, cranial nerve 9, and a branch of the facial nerve, cranial nerve 7, known as the chorda tympani. So all of these different nerves will make contact. They are cranial nerves, so they will go to the brainstem. They will then make contact with the nucleus of the solitary tract. Now, because this is taste, it does actually make contact in the thalamus. If the first place it will go to in the cortex after making contact with the thalamus is what we call our gustatory cortex, which is a portion of the insula, and then finally making additional contact in the orbitofrontal cortex, which we also heard about when we talked about our sense of smell. So our main sensory receptors are the taste buds. Now, when I was growing up, I thought, and if you take a look at your tongue right now, you'll kind of see that it is covered in a variety of different bumps. Those bumps are not taste buds. Taste buds are actually located inside of those bumps. So taste buds basically will create neural signals, so they'll engage in that process of transduction that will be sent to the brain via those taste nerves that I showed you. They are embedded in papilla, and the papilla are those bumps that are on your tongue, and these contain our gustatory receptor cells. So there are 4 different types of papilla on our tongue. Each of these papilla will contain, depending on the size of the papilla, a certain number of taste buds. So the first ones that you'll kind of notice, they're not here in the picture, but they do contribute to the overall bumpy appearance of our tongue. They're not in this image though. They're called filiform papilla. These are what give your tongue a bumpy appearance. They don't actually have any taste function whatsoever. The 3 other types of papilla that we're going to talk about do actually play a role in taste function. So the first ones that we're going to talk about are fungiform papilla, which are located on the edges and on the tip of your tongue. Your book mentions that you can very easily see your fungiform and filiform papilla by basically getting some blue food coloring or like eating a blue popsicle. It will stain both the filiform and the fungiform papilla. The fungiform will be a little easier to notice because they will tend to have a lighter color than your filiform papilla. So generally with our fungiform papilla, they have about 6 buds per papilla buried under the surface. Then we have our 2 other types of papilla. So here we have what are called our foliate papilla. We call them that because they are folds. So we have these folds of tissue, and within those folds, we have taste buds. And you can kind of see here from this image, the foliate are located in the rear and they're on the lateral surface of the tongue. And then finally, we have the circumvallate papilla. These are very circular. As you can kind of see, they have a mound structure that's kind of surrounded by a trench. And all of these 3 different types of papilla will provide functions with respect to taste because they do contain taste buds. Now, let's talk a little bit more about taste buds and taste receptor cells. So a taste bud itself is made up of groups of microvilli, which you can kind of see here, and taste receptor cells, sensory cells. So here we have some taste bud sensory cells. We also have some microvilli. Microvilli are slender projections that occur on some of the taste bud cells, and they do extend into this little opening that's called the taste pore. The microvilli have sites that basically bind to tastants. So when different molecules of taste enter the tongue and enter the papilla, they're going to make contact with the microvilli that extend out into this pore. We didn't really know what microvilli were for a very long period of time. We now know that they are actually extensions of the cell membrane. So from the microvilli, in addition to that, we have our taste bud receptor cells. And basically, we have about 3 different types. So we have one type that specifically codes for salty, and that's going to focus on the ion channels that are opened, things like sodium, which does have a bit of a salty flavor to it. So type 1 is often described as having a bit of a housekeeping cell. It excretes potassium through the taste pore, and this basically keeps the resting membrane potential of the other cells in that taste pore stable. Type 2 are true receptor cells. They respond to sweet, bitter, and possibly umami tastes. And then you'll also kind of notice they do have some G protein coupled receptors, but they don't actually have synapses, as you can see here, and neither does type 1. Type 3, which seems to code for sour taste, does have a true synapse. And they are actually presynaptic, so they do actually converge onto nerve fibers. And it's believed that these might play a role in that sour taste. So tastants, just like we talked about odorants, tastants are any stimulus that can be tasted. And we tend to divide those into 2 different types of classes. So they can be composed of these small charged particles that basically taste either salty or sour. And this is largely because of ion channels opening. We perceive these through ion channels in the microvilli. Or for something that's a little bit more complex, like sweetness or bitterness, we perceive them via G protein coupled receptors. So after taste information goes to the taste buds, all of the gustatory cells basically form a gustatory nerve, and that information will proceed from those different cranial nerves that we mentioned before making contact with the medulla and the thalamus. And then after that, it's going to reach the cortex. So the first place that it reaches is the insular cortex, or the gustatory cortex. And this is the primary cortical processing area for taste. Now remember that the insula is responsible for a lot of different things, not just taste. Now we have retinotopic maps, we have tonotopic maps, we have somatotopic maps. Do we have a gustotopic map? Originally it was thought that we didn't, but it does seem that the insula is organized into different clusters that are based on taste type, so we might actually have a gustotopic map. After making contact with the insula, information will be sent to the orbitofrontal cortex. This is an area that's responsible for integrating information with senses, so it will also take into account the other sensory experiences of taste, things like temperature, touch, smell, in addition to taste. So again, you can kind of see how this works. So we have our 3 cranial nerves, 7, 9, and 10, making contact with the nucleus of the solitary tract and the pons and medulla before going to the thalamus. So here we can see our thalamus, then it goes to the insula before making contact with the orbitofrontal cortex. Now let's talk about our 4 basic tastes. Can you name them? Odds are pretty good. If you've been listening to this lecture, you can. So we have sweet, salty, sour, and bitter. So let's talk about each of these in turn. So your sense of saltiness comes from salt in food. So a tastant must have salt to kind of have a salty taste. Any salt is basically going to be made up of two charged particles. It's going to be made of a cation, which carries a positive charge, and an anion, which carries a negative charge. So for example, if we're dealing with standard table salt, which is sodium chloride, sodium is our cation, chloride is our anion. Sodium carries a positive charge, chloride carries a negative charge. Your ability to perceive saltiness really fluctuates. And one of the ways that you can kind of see this is if you change your diet. So if you decide to go on a diet that's very, very low in sodium, what you're going to find is the things that you used to not find very salty will all of a sudden become very, very salty to you. So if you don't have a lot of salt for a while, things that weren't salty to you before will suddenly become very, very salty. So your ability to perceive saltiness can change based on the input that you're providing your system. Additionally, we know that liking saltiness can also fluctuate. Now, I know for me, I love salty snacks. And there's been some research that suggests that perhaps people tend to want salty snacks when they're dehydrated. One of the things that we do know is that children that had chloride deficiencies in childhood might develop a preference for salty foods later on. Additionally, we know that mothers that have severe morning sickness, things like hypermeiosis gravis, during gestation, we do know that children that are born to mothers that have that severe morning sickness may also have a craving for salty foods that children with mothers that don't suffer from that morning sickness really don't. And that, again, could be due to nutritional deficiencies that developed in utero. The sour taste. Now what you're looking at here are mega sour warheads. That is really my first big experience with sour things. My favorite candy is Sour Patch Kids. But when I was growing up in the 90s, I loved me some sour warheads. We actually had somebody in our geometry class that would always bring warheads to class. So geometry, even though it was my worst class, was also my favorite class. Sour taste basically comes from acid. And so what this means is that for something to have a sour taste, it has to have a pretty good concentration of hydrogen ions. A little bit of sour is okay. So if you like lemonade, that is okay. If you like sour candy like I do, that's okay. But we do know that at high concentrations, acids can damage internal and external tissues. The bitter taste. So I'm not a big fan of black coffee. Black coffee can be very, very bitter. Probably the prototypical bitter tasting substance is quinine, which is present in tonic water. Quinine is incredibly bitter. It was previously believed to be a good treatment for malaria. It really isn't. You might have heard of quinine as a potential solution in the 1800s and 1900s to get kids from biting their fingernails. It is so bitter tasting that the only way to really have it in people find it so unpleasant that tonic water actually has a ridiculous amount of sugar in it to cut that bitter taste. The sugar content in tonic water is actually pretty similar to what you'd find in a regular soda. That's how much sugar you need to actually cut the bitterness. And even then, even with all that sugar, a lot of people don't like tonic water. I'm one of those weirdos who does. If we were having class right now, I would have actually brought in some tonic water and you could have tried it for yourself. And odds are pretty good you wouldn't have liked it and I would have finished it off at the end of class. Generally, with bitterness, you and I can detect different types of sour tastes. You and I can detect different types of sweet tastes, but we can't actually do a very good job of distinguishing between different types of bitterness. The general rule for most of us is that if it's too bitter, we're not going to try it. And part of the reason for this is that bitter taste is often a signal for poison. Oftentimes poisons have a very bitter taste. Having said that though, some people really like black coffee, and some bitter tastes are actually really beneficial. Bitter taste can be present in certain types of vegetables. That's especially true for certain things like spinach, collard greens, Brussels sprouts, and certain types of broccoli. So the bitterness is there, but in this case, the bitterness is beneficial. Those bitter flavors signal things like minerals and vitamins that can protect against different types of cancers. Then we have the sweet taste. Of course, this is evoked by sugar, and there are different types of sugars out there. They all taste sweet. So we have glucose, which is our primary energy source. We have sucrose, which is your standard table sugar, which is made up of a combination of glucose and fructose. And then we have fructose, which is made up of natural fruit sugars and is generally sweeter than glucose. All sweet perception is determined by a single type of receptor. It is a metabotropic receptor. I'm not actually going to show it to you. It's a little terrifying to look at. But different sweeteners, including artificial ones like sucralose or aspartame, activate different parts of the receptor, but they all work on the same type of receptor, producing that sweet taste. So now let's talk a little bit about the variability that we see in taste. So we got to go back to the very beginning of class when I had you test whether or not you were super tasters. So what you are looking here is phenylthiocarbamide or PTC. So Arthur Fox did some research. He's a researcher, and he basically reported that PTC, this chemical structure here, tastes different to different people. And this is just my reminder to you that when we did the super tasting, that taste strip was actually coated with PTC. And when people taste this substance, some people detect a bitter taste, some detect it immediately, some take a little more time, and other people taste nothing. Now, a very similar bitter substance that is often only detected by super tasters is what is called propylthiouracil, or PROP. And researchers were actually able to isolate the gene for PTC or PROP in 2003. So if you have two recessive genes, you are what are known as a non-taster. So if you didn't taste this, you probably carry two recessive alleles. If you have one dominant allele, you're probably going to be a medium taster. You're somebody who can taste the bitterness, but it might take some time. If you have two dominant alleles, you're probably going to be more of a super taster. But I want to be very clear. Just because you have these alleles does not mean that you are specifically a super taster. We actually have very specific requirements for what you need to be able to be a super taster. But what this does tell us is that some variability that we have in taste. Some of us like certain foods and some of us don't. I cannot handle overly spicy food. Now, some of that might be a product of my upbringing. Some of that is probably also my genes and my bland Scottish heritage. But this kind of shows that the variability we see might have a genetic component. So what does it take to be a super taster? You do need to be able to taste PTC or PROP, but you also have to have a very high density of fungiform papilla. So here's an average taster. So you can actually see the fungiform papilla. I'm kind of circling them with my cursor right now. This is an average taster. They've got a pretty good amount of fungiform papilla. Take a look at our super taster who has significantly more fungiform papilla. So super tasters will perceive the most intense sensations. In addition to super tasting those bitter tastes, they will perceive greater oral burn from spicy food. They will also experience oral touch like fats and food thickeners. So they'll be super duper sensitive to certain types of food textures in a way that an average taster or a non-taster might not. So what are the consequences of taste? Well, the variation that we see in individuals and foods will potentially affect preferences, and it will also affect diet. Now, I am probably an average taster or a medium taster, but if I were a super taster and super sensitive to bitterness, I may not like vegetables as much. In fact, we do tend to find that people who really can taste that bitterness may not eat as many vegetables because, well, those are more opportunities to encounter that bitter taste. Now, because super tasters are sensitive to that bitterness, they may not eat as many vegetables, but they're also potentially going to eat fewer high-fat foods because fat changes the texture of foods, and because they can taste the richness of the food, and they may eat fewer of them. Now compare that to somebody like a non-taster who's not really sensitive to that bitterness. There's some evidence that they're a lot more likely to smoke because that bitter flavor doesn't really bother them, and drink alcohol.